Okay, so hello and welcome to Excelsior Academy. So guys, till now we have discussed like how we can start the isolation and purification of RNA and DNA, right? So till now what we have discussed is where exactly to start. Okay, so we know how to start now, and what we are discussing right now is all the conventional method. Now, why we started with conventional method, even though in company, in the research lab, we don't maximum time we don't use conventional method. We go with the kit, okay, and these kits are designed by the companies. So even this I'm going to discuss so that you are familiar with like what kind of method is used in the research lab. And this conventional method I'm discussing so that the theory or the basic concepts behind the isolation and purification is clear. Okay, so today we'll be completing the conventional method, and then next lecture we'll be doing the kit method. So by this two methods you will be like clear. Okay, so the theory, you know why we are doing such step, like for example why we were using phenol, why chloroform was used, why alcohol was used. Okay, so now you know why these chemicals were used. Now, in the case of kit, they are not known as phenol, they are not known as chloroform or absolute alcohol. What kit is having is like solution 1, solution 2, and they will not tell you which one is what. That is like, you know, patent, and they will not reveal. But you should understand that first thing is, of course, the phenol kind of thing it will be. Second will be chlorophenicol or alcohol. So like this, you will understand the steps. Okay, so the basic concept जो रहेगा वो कोई change नहीं करता. It's just that they won't share the name and what are the ingredients which is present. So in the previous lecture we have discussed like how at the end you are going to get the DNA in the pellet. So if you remember, we have discussed that once you add alcohol, okay, and I have mentioned that you should use cold isopropanol, right? So at the end, in the appendor, okay, you will see that the DNA is present. And I have also mentioned that this should be reconstituted. What is the word? It is a reconstitution. Okay, so it should be reconstituted either in double ionized, deionized water, or millicube we say. Why we say millicube? Because it's the name of the company which gives you the deionized water. Okay, तो short form में क्या होता है? Whether you have got millicube or not, हम ऐसे नहीं बोलते do you have double deionized water, वो थोड़ा लंबा पड़ जाता है. So slang कैसे हो जाता है लैब में कि आपके पास millicube है या नहीं है. Okay, it is a company which gives you the it's a machine, okay, which gives you the double deionized water. Okay, so either in the millicube or in the TE buffer. Okay, so I have mentioned what is TE buffer. It is your tris EDTA buffer. Okay, वो भी कितना आप ऐड करोगे? Around 20 to 50 microliter, not more than that. Okay, so Chandni, what will happen if I add 100 microliter? What will happen? Like why we are stuck only to 20 to 50 microliter and why not 100 microliter? Especially in the case of bacterial DNA. But in the...
Can you repeat it again? My connection lost, ma'am. I have joined now.
Okay. So what I was saying is that at the end, when you reconstitute DNA, you use either millicube or tris EDTA buffer around 20 to 50 microliter. Okay, but we don't add 100 microliter or 500 microliter. Why don't we do this thing? Like why can't we add 500 microliter? Anyone can answer. It's okay. So Chandni, what will be the answer? Why are we not using 500 microliter, but instead of that, we should use 50 microliter? What is the logic behind?
To easily isolate, ma'am.
No. See, your DNA can come in 50 microliter also and in 500 microliter also. But I'm saying that when you do a research, you should add 50 microliter only max. Okay, out of which, as I mentioned, you will take 1 microliter and go to nano drop, where you will check the concentration of DNA. Okay, and then based on the concentration of DNA, you will have your own experiment. What if I add 500 microliter to my sample? What will happen? And why it is a wrong procedure? Akshaya, do you want to try? Okay, yes, Chandni. So what was your answer?
It will become difficult to absorb, ma'am, clearly.
No. Okay, anyone else would like to try, Tanvi? Okay, before I proceed, the question is clear to everyone, right? The question is clear. So you remember the last step?
Yes.
Okay. So Shruti, why you think that we shouldn't add 500 microliter and why 50 microliter?
Well, it interfere with the properties of the extracted genetic material because it's a chelating agent. It might do something to the protein which is extracted. No? Okay.
I have already given the hint. At the end, you are going to take 1 microliter of sample, correct? And go for DNA concentration check. What happens when you add more of the buffer and less? क्या होता है? Tanvi, do you want to try? Okay, I'll give you the answer. Yes.
Will it interfere with the reading?
Not exactly. But you are on the right track. Can you try more? It does not interfere with the reading, but it does something to the reading.
Ma'am, will it dissolve the DNA?
It should, right? It should be soluble in the buffer. तभी तो मैं आगे काम कर पाऊंगी, right? So what you get at the end is the precipitate, DNA precipitate, because alcohol, it is not soluble, right? Alcohol क्या करता है? It is a dehydrating reagent, right? What is the chemical nature? What is the property of alcohol? That it dehydrates. So you get a DNA precipitate at the end. But I cannot work with the precipitate. So I will put into the water or the TE buffer so that I can get access and I can work. Precipitate पे enzymes काम नहीं करेगा. It should be soluble. So it is okay. I mean, it's correct that it should get dissolved. Dissolve मतलब गायब नहीं होता ना. It just gets reconstituted in water because in the nucleoplasm also your DNA is suspended. जो भी nucleoplasm रहता है, it is not like ऐसे cavity type where nothing is there. Correct? You do have the nucleoplasm and cytoplasm. Okay, I'll give. अभी जो मैं आपको आंसर दूंगी ना, you'll be like, oh my god, this was the answer type. ऐसा होगा. Okay, so the question was from the appendor, you have got the DNA over here. Okay, now I have added 50 microliter and took 1 microliter, gone for DNA reading. Okay, so you know that DNA reading is at 260 nanometer. This is clear. I can do the same thing with 500 microliter of TE buffer. Again, take 1 microliter and go for DNA reading at 260 nanometer. अब मुझे ये बताओ, in among these two scenario, let us say that this is A scenario, this is B scenario. Among A or B, which one is going to give me high concentration of DNA as reading?
B.
B.
Correct. Correct. So what is happening when you are adding more buffer? Your DNA is
the concentration is lower.
Yes, your DNA is
And
getting.
therefore the reading. Okay. Yeah,
Now
that
what
is how the
happens?
reading was affected.
So this is how the reading is getting affected. Now see, answer कितना simple था. आप तो बहुत ज़्यादा complicate सोचते हो. इतना नहीं जाना है. So it gets diluted. Correct. अब क्या होता है in later stages when you're doing mole biocloning and whatever restriction enzyme digest या जो भी whatever work you do, you need to carry out everything in 20 microliter of total reaction. Okay, total reaction. Now, if I have diluted reading, suppose I'm saying that it should have 100 nanogram of DNA concentration in order to start the PCR or whatever you are doing. Okay, this 100 nanogram should be in 2 microliter of the total reaction, क्योंकि total 20 microliter है, जिसमें से 2 microliter आपका DNA template होगा, कुछ फॉरवर्ड रिवर्स प्राइमर होगा, कुछ MgCl2 होगा, कुछ बफर होगा, कुछ माइक्रोलीटर आपका DNA पॉलीमरेज होगा, राइट? सो टोटल इट शुड बी 20 माइक्रोलीटर. ये कब पॉसिबल है? when you have got the high concentration in your stock solution of your DNA. But if the stock solution is diluted, then in order to get 100 nanogram, I need to increase. Maybe I have to take 30 microliter so that I get 100 nanogram of DNA concentration. But then I cannot make PCR reaction which is about 20 microliter. Correct? अभी समझ में आया?
Yes, ma'am.
Okay, yes, it was simple. So that's why we add less of a DE buffer. Okay? Now, see, it's not like it's ऐसे कहीं पे पत्थर पे लिखा नहीं है that it should be always 50 microliter. Now, when you can go for 100 microliter, I'll tell you that also. So you know that as compared to bacteria, as compared to animal, the plant DNA is much more correct. अगर मैं 100 ग्राम बैक्टीरिया कल्चर ले रही हूँ एंड 100 एनिमल ले रही हूँ, आई एम गोना गेट मोर इन द केस ऑफ प्लांट. ठीक है, क्योंकि उसका एक्सट्रैक्शन थोड़ा इजी हो जाता है. सो प्लांट डीएनए आई गेट मोर. नाउ एट द एंड आई गेट द प्रेसिपिटेट एंड आई एड if I add only 50 microliter, what will happen is that the 1 microliter solution, okay, of DNA concentration at 260 nanometer, the reading is going to be beyond the standard curve. अब standard curve क्या होता है? That एक इतना है तो इतना नैनोग्राम है. If this is the absorbance at 260, then this is the nanogram concentration. But what will happen if it is falling, say, out of the standard graph? Means the reading is very high and it is not falling in the standard graph. So then I don't know, because if the reading is coming around 4 OD at 260 nanometer, ठीक है, but मैंने तो सिर्फ OD1 तक ही बनाया है. For example, 0.02 OD means इतना concentration, 0.04 OD means this much concentration. ऐसे छोटा छोटा करते करते सिर्फ 1 OD तक मैंने बनाया है, where I can read around 100 nanogram of 100 nanogram of DNA. Correct? This is my standard graph. Now, if I'm getting OD4, it means what? It is falling out of my standard curve. Correct? Is this much clear, guys? ये समझ में आया?
Yes, ma'am.
Yes,
now,
ma'am.
since it is high, okay, 50 से काम नहीं चल रहा है, then I'm gonna add 50 more. So right now, what I've done, I have diluted 1:1. ठीक है? मैंने 1:1 का dilution किया है. Total now this has become 100 microliter. Again, I'm gonna take 1 microliter, go for 260 वाला reading. So maybe 4 OD, say this had, might have become 1.5 wala OD now. Still it is falling out. Correct? It is not falling in the standard curve. Again, what I'm gonna do? फिर से I'm gonna add another 50 microliter. That time शायद 0.8 में आ जाए. So 0.8 will then correspond to me कि इतना इसमें present है. Then I should calculate कि हाँ. So I have added 150 microliter total, in which 1 microliter means this much of nanogram. So 150 is going to have this much nanogram of DNA. Is calculation clear? अगर मुझे पता है 1 microliter में कितना है, so I can back calculate for 150 microliter of total DNA solution. Correct? Simple?
Yes, ma'am.
Yes, ma'am.
Cool. So let's go ahead. So now, so this was all about, like, you have got the DNA. Now there are certain techniques, okay, by which you can also purify DNA. So that is what we are going to discuss. Again, ये थोड़ा सा complicated हो जाता है. When and why to use, उसमें आ जाता है. How to means it's okay. You can go to the lab and you can run. So right now we are going to discuss about the centrifugation, gradient centrifugation. So you know what is centrifugation. Now there are 2 types of centrifugation. One is gradient and one is isopycnic, out of which for separation of DNA, we need अपना gradient centrifugation. And here we don't purify DNA. What do we purify is nucleic acid. Sorry, nucleic acid बोल रही हूँ, nucleus. अंतर समझ में आ रहा है? When I say that I'm going to purify nucleus rather than nucleic acid. Yes? Any confusion with the word? Okay, let's go.
Ma'am, why we have to isolate nucleus itself, ma'am?
See, that's what, na? There are different ways. If I wanted to work on the nuclear protein only, then I will not disrupt the entire cell. See, simple. Suppose I am interested to work on the nuclear protein. Okay. So if I rupture the entire thing, okay, and the DNA, RNA, everything comes out. So this nuclear protein which is present, okay, this is cell, this is nucleus. So the proteins which are present over here during the purification will get mixed with the cytoplasm, it will get mixed with the nucleoplasm. So the load of purifying protein will increase. Correct?
Yes, ma'am.
So what is the best method? That I go for the nucleus purification. This is one example where you can use gradient centrifugation. So I'll go for nucleus purification. Then I will add detergent so that only the nuclear membrane proteins will remain active and nucleoplasm protein will precipitate out. So right now I'm just having all the nuclear proteins. Then later, whichever protein you're interested, you will just purify depending on the different types of chromatography techniques. Are you guys able to follow me or getting confused?
Yes, ma'am.
Okay. So let's continue with what is the gradient centrifugation and when and how to use. So centrifugation at moderate speed is frequently used in DNA purification and separation, whether it be removal of debris or recovery of the precipitated nucleic acid. So just we have seen in the previous lecture. It is also frequently employed in various column purification. So what is column purification? So when you say column purification, आपके दिमाग में क्या आता होगा? That there is a column, right? It will be eluting out and you will be collecting. But when you say column purification, the company has made ऐसे 1 centimeter का column. And this column is fixed into the centrifuge tube. Okay. If I say that this is centrifuge tube, तो आपका column कहाँ आ रहा है? This is a column over here. Okay. So now what you're doing is you're adding your sample. And whatever is required, that will get bind to the column. Rest all will come out. It will be eluted out. This is the terminology we use. Okay. Next, what I'm going to do is I'm going to add the elution buffer, which will remove the required sample. So then I will add, that is called elution buffer. Okay. So this elution buffer, after this, the debris which I don't want, it's removed. Another, I will add this elution buffer and it will come out. So in the next round, whatever comes over here, it has got my interested sample. Okay. This I will collect and start doing my experiment. Clear or confused? Should I repeat?
Yes, ma'am.
It was clear.
Okay, I didn't get. Repeat or continue?
Continue, ma'am.
Okay. So this is one method. Okay. So let's just see how the gradient centrifugation is helping. So it is employed in various column purification methods. So apart from these methods, there is another old method of separating nucleic acids or any type of macromolecule, and that is your density gradient centrifugation. So that is what we are going to discuss. Now, there are different types of gradient centrifugation. So we have got sucrose, we have got cesium chloride, okay? and the different are there for like where you can use ethylene bromide for the separation of plasmid DNA, etc. But mainly we use sucrose and cesium chloride. Okay. So in addition, sucrose gradient can be used for size selection of large DNA fragments when you are constructing a genomic library. And in this case, you can also separate supercoiled versus relaxed. So what is relaxed? Relaxed is your linear DNA. Supercoiled is the DNA which is like this. Remember, it is also called triple C, covalently circular closed circular DNA, right? So plasmid cases mein you have this. So what happens? These are very tightly and these are very relaxed. So because of this, again, there is a difference in the density, and hence you can separate two different types of DNA. Okay. I'll show with the figure also, don't worry. So this is your sucrose gradient we are going to discuss now. Okay. So you take a you take a centrifuge tube. You don't take test tube. Test tube क्या है? कांच का रहता है. It will break under the centrifugal force. So you are going to take a plastic का made centrifuge tube. Okay. Now what you do is, see, can you see the gradient? It is 80%, 35%, 15%. And depending on your requirement, you can have the difference in the density of sucrose. Simple. Ab 80% sucrose hai, what does it mean? How you are going to prepare sucrose 80%? Kaise banaoge? If in the lab, I am your supervisor and asking you to prepare 80% sucrose solution. Okay? Let us say that 20 ml. Here you see, you can see that it is they are just adding 100 microliters. So 20 ml bhi bahut zyada ho gaya. So let's say that you have to make 5 ml of 80% sucrose. Kaise banaoge in the lab?
We make 80% of 5 ml. Like we have to calculate like kitna hai. Okay, 80% of 5 ml is about 0.4 microliter, I guess. And then the remaining will be water.
Correct, but see, when you say percentage sucrose is not liquid. It's a
we have to weigh.
So you have to take the weight. Okay. So जब हम 80% sucrose बनाते हैं, so how you are going to take 80 gram? It means 80 gram in 100 ml of water. Okay. So I have asked to make 5 ml. So simple calculation. यहाँ पे 5 आने के लिए what you can do is you can divide by 20. Correct? Sorry. So 5 ml आ जाएगा यहाँ पे. If you divide 80 by 20, how much you are getting? 4 gram. Am I correct?
Yes, ma'am.
Okay. So 80 gram in 100 ml is nothing but your 4 gram in 5 ml. That is your 80% sucrose solution. ठीक है? अब कैसे करोगे? Practically कैसे करोगे? So you're going to weigh 4 gram. You're going to put into at least 3 ml. Okay. And make up the volume to 5 ml. Out of which 5 ml you are going to take 100 microliter. Okay. This is your first step. Now, what is the second step? Similarly, you are going to make 35% of sucrose. Add 200 microliter of your sample. अब कैसे add करोगे? That is also very important. So, you tilt the test tube. Okay. You tilt the test tube and slowly you are going to add. So that what happens? अगर आप ऐसे mix कर दोगे forcefully, ऐसे pipette से डाल दोगे, so what will happen? This 80% and 35% mix हो जाएंगे. Then total resulting percentage will be different. So slowly add. So to touching the wall of the test tube, you are going to put. Okay? So then slowly you will see that there is a 80% ka gradient. Then you have got the 35% gradient. Okay. Similarly, third step is 15% sucrose, 200 microliter. Now on to top of this. Now on the top of this, you are going to add 100 microliter of your sample. अब ये कौन सा सैंपल है आपका? जो भी आपने होमोजेनाइजेशन के बाद का है, या प्लांट डीएनए का लाइजेट है या बैक्टीरियल सेल लाइजेट है. ठीक है? दिस इज योर सैंपल. नाउ यू आर गोइंग टू सेंट्रीफ्यूज फॉर 1 hour, जो भी आपको आरपीएम दिया है. Now, one more thing, this is not fixed. The addition of 200 microliter, 100 microliter, it's not fixed. Okay, it varies according to the requirement of the experiment and from lab to lab. But for your understanding, I'll tell you, हम 100%, okay, 100 तो doesn't work, so 90 है, 90% है, people are there who make 80% also, 70% also, 60% also, like that. So depending on the requirement. But please do understand, you get a gradient like this. Okay, is this part is clear to everyone?
Yes, ma'am.
Okay.
Yes.
Okay. So yeah. So now you put into the centrifuge for 1 hour and you give the RPM. Okay. What will happen from the sample? You can see over here. At this region, you have got all the membrane organelles which is coming and concentrated over here, where at the junction of 35 and 50% density. So it means what? It means that the membrane organelles like mitochondria, then you have got different organelles like chloroplast, if suppose it's a plant one, or you can have what else is there? You have got what else is peroxisomes are there, vacuoles are there. It means they are getting concentrated over here because their density matches with the density over here of the sucrose. Clear? And the soluble proteins, only soluble proteins are there which is present at the top. Clear? Now, if suppose Akshaya wants to work on the membrane organelles, so what she's gonna do? She is gonna harvest, okay, this portion. So this she is gonna take and throw, and only this portion she is going to be interested, and so that later she can work on the mitochondria, for example. Okay, agar they had nucleus also, okay, nucleus also. So nucleus will come at the bottom, wherever the density is maximum. So nucleus will be here. Okay, is it clear? This is how your sucrose or cesium chloride density based gradient centrifugation works. If you're interested to work upon the organelles, intact organelles, okay, here you will get intact mitochondria, intact chloroplast or peroxisome or vacuoles, whatever. And this is how you get the intact nucleus also. Clear? Is this much clear?
Yes, ma'am.
Okay. So this is how, can you see? So there are two are there. So this is one centrifuge. Can you see? It's actually from the, I mean, looking at this cover itself, I can say that this belongs to Tarson company. Okay, so they have got the blue cap and this white area. Can you see? This is meant for labeling, so that you label what you have added into this, and one you keep it for balance. So in the case of centrifuge, you need to have a balance. If suppose it's not properly balanced, okay, if it is not properly balanced, the machine is not going to start. It will not, start rotating. It will not. So this is how you keep, okay, your machine looks like this. Now, so look it over here. So you had difference in the densities. And of course, they are your sucrose doesn't have any color, so you won't see this much. But you can label this. Ki maine 200 microliter 60 dala, 250 dala. Like this, you can label on your centrifuge, and this is your sample. Okay. After centrifuge, what do you see? Can you see the gradient difference? Can you see? Right? So depending on this, you can see this is clear portion. This is little light portion. This is again another portion. This is little dark, and again here. So less density. So I'm gonna harvest using the pipette into different appendogs. And later I can do the experiment to confirm whether I have got the mitochondria or ribosomes or proteins or nucleus. Okay. So this is how your gradient centrifugation works, and which helps in the purification of the organelles, mainly organelles based on the density. Yes, is it clear? Everyone, any part where you think that you want me to repeat or it is not clear? Everyone, it's clear?
Yes, ma'am.
Yes, ma'am.
Okay. Okay. So now coming to the alkaline denaturation. So this is mainly used, okay, this is mainly used for your plasmid. Okay, so now we are going to like see why and how this alkaline denaturation is used for the plasmid. And there is one book called Sambrook, if you have heard of it. So they have got this volume 1, 2, and 3. There are 3 books are there, and very beautifully they have given each and every step and why that step is being done, okay, in order to purify plasmid or any other DNA. So if you guys, get a chance to go through this book, it's called Sambrook Molecular Cloning. The title of the book is Molecular Cloning, okay, written by Sambrook, volume 1, 2, 3, and very nicely they have given. So worldwide, like wherever the research is being done, we all follow this procedure, okay. Now, in a typical extraction and purification procedure for plasmid DNA, bacterial cells with the desired plasmid are lysed under the alkaline conditions, and the crude lysate is purified using the filters of the centrifugation. Again, so it is the lysate. Okay, lysate क्या होता है? जब जैसे ही आपने cell wall break किया, whatever comes out, okay, it is called as lysate. So it is loaded onto the apparatus where plasmid DNA binds under appropriate low salt and pH conditions. So these are the points which you need to remember. So plasmid DNA is done for the plasmid DNA is done under the alkaline conditions, and it also requires low salt and pH conditions. Okay, so these are the points which they always ask in the MCQ. RNA, proteins, metabolites, and other low molecular weight impurities are removed by medium salt wash, and then the plasmid DNA is released in the high salt buffer. And then later it can be concentrated and desalted for the genetic engineering uses. So what does it mean? See, the thing is that already we have done a lot. So here, thoda direct answer diya hai. So what you're doing is you get the lysate, you put into the column. Okay? So in the column, see, this is how. Wait, let me draw it properly for you guys. So this is how your column is. Yeah, आपका cap हो गया. Here, this is your column actually. Okay, append में ये column रहता है. So you added the lysate. So this column is designed in such a way that only the plasmid DNA should bind. But sometimes the associated proteins, the associated metabolites will also get stuck. Okay, so that will be removed when you give a wash with the medium salt or low salt conditions. So उसमें क्या होगा? ये छोटे-छोटे जो impurities हैं, that will be washed off. Now you want to purify plasmid DNA. So plasmid DNA will be eluted using the high salt buffer. Okay. Now, what is the chemistry behind using high salt buffer? Can anyone tell me? Anyone?
Neutralize.
Huh? To?
Neutralize the DNA.
No, it's not for the neutralization. It's if you know the high salt concentration, they are good in breaking all kinds of electrostatic interactions. Okay, so जो भी column बनता है, it is again based on the electrostatic interactions because the DNA, it has got the phosphate group, correct? So of course the DNA is going, the column is going to have some group which will be having the basic charge, the positive charge, so that it is able to bind with the negative charge of the DNA. Now, when you add high concentration of salt, what it does is it breaks all those interactions, and hence it is able to elute out the DNA. Okay. Okay. Now the thing is that this eluted DNA is going to have high salt concentration, which is not good. If in future I'm going to do cloning, the enzymes will get precipitate out if I have high salt. So I will do the desalting. And for desalting, again, you have got different columns for this. So that is going to remove all your salt and only DNA will be present. Okay. So far, so good, guys. Able to follow? Yeah.
Yes, ma'am.
So in bacterial cell extract, chromosomal DNA exists as a linear fragments due to fragmentation which occurs during the lysis of the cell. ठीक है. So raising the pH to about 12, it will disrupt the hydrogen bonds and allow the linear strands to separate. Plasmids are tougher, so they are not disrupted by the cell lysis and they will remain intact as the supercoiled circular DNA, as I showed below. ऐसा कुछ, ठीक है. So what you purify is this. So although the pH will disrupt the hydrogen bond, the two circular strands will not be able to separate physically and it will be interlinked. So इससे क्या होगा, आपका plasmid DNA जो है वो intact रहेगा, whereas the other chromosomal DNA will be disrupted. And moreover, in this method, we are interested in the plasmid DNA. Okay. So when the pH is reduced, the interlinked plasmid strands will snap back to form the double-stranded plasmid in case if they get separated. On the other hand, the linear chromosomal fragments cannot do this. They get aggregated. It becomes an insoluble fragment and they will be removed during the centrifugation. So they will just precipitate out. Now look at the figure over here. So you have got the chromosomal DNA as the linear fragments. Why? In the bacteria, I know that the chromosome is circular just because of the shearing or the mechanical forces which you use for the cell lysis, which results in the chromosomal DNA break, and that forms a linear DNA, whereas the black one is your plasmid DNA. Okay. Next, denaturation, they get separated. You can see the strands are getting separated. Okay, the hydrogen bonds are broken. So denatured plasmid DNA strands remain interlocked. Can you see? It is interlocked. They are not broken totally. And they are dissociated, the linear strands. Then on removing the renaturation, on coming back to the renaturation, you have got your interlocked plasmid, and hence you can purify plasmid easily as compared to the chromosomal DNA. Are you able to follow this figure? Yes? Okay. Now coming to the column purification. So there are two types of column purification for the nucleic acids. So in size selection chromatography, a sample is passed through the matrix of small pores. So just get the idea over here, because in the upcoming techniques, I'm going to teach you in detail how the size selection chromatography or affinity chromatography works. Okay. So just get the idea that size means it is based on the size and the shape of the molecule. Okay. So smaller molecules like salts or the nucleotides, they will enter the beads. So ये क्या होता है, size exclusion में you have got the beads like this. Okay. So if it is small, if the sample is small, it will enter the beads because it has got the pores. So it will enter the beads, then from here it will enter here, from here it will enter another beads. But if the size is big, it will not enter, but it will pass through the gaps which is present between the beads. So tell me, which will come out faster? Which is crossing the beads or which is going through the beads?
Crossing the beads.
Crossing the beads one.
So जो आपके beads के बाहर से cross कर रहा है वो आपका सबसे पहले elute out होगा. So you know that you have got the longer nucleic acid, so they will just pass through the column. Okay. We'll come to this. This is also very important. Okay, so many numericals is also based on this size selection or the size exclusion chromatography. It is also known as gel permeation chromatography. Many names are there. Now coming to the affinity chromatography. So affinity means they are going to be specific. Okay, so this could be your anionic resins which binds to the negatively charged phosphate groups. So when you say anionic resins, इसका मतलब क्या है, वो negative charge को add करेगा. It means the matrix is having the positive charge. ठीक है? Or you can also have resins which are coated with oligo DT sequences. अगर उसके पास in the bead it has got a long stretch of T. So in your knowledge, which has got the long stretch of A. So if you know mRNA, it has got a poly A tail. It has got the 5' cap and 3' poly A tail, correct? So if I want to purify mRNA, then I can use such resins which has got oligo DT sequence. So they can bind to the mRNA molecules. Clear? So this is the example of affinity. यानी कि मैं सिर्फ एक तरह की चीज based on the affinity to each other I can purify. Clear guys? Just get the idea, because this all I will take in detail, don't worry. Okay, now the important point is about the detection and the quantification of nucleic acids. Okay. Now, so we know that we are going to quantify DNA or RNA at 260 nanometer. And you should also know that why it is at 260 nanometer, because the heterocyclic bases are having the absorbance at this particular wavelength. Okay. Now, this is convenient but not very sensitive. A solution of 50 microgram per ml of double-stranded DNA will have absorbance of 1. But we don't prepare 50 microgram per ml. What will be the shorter form of this? Oh, sorry, मुझे और confuse हो जाएगा, sorry. I should write this in the nanogram. So nanogram के लिए कैसे करेंगे? So this is -6 means microgram. This is gram. For nanogram, I should divide. This is 10,000. Okay. 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 So 50 microgram per ml is also 50 nanogram per microliter. Is the calculation clear to you? मैंने क्या किया यहाँ पे?
Yes, ma'am.
Okay, okay. So this is nothing. So we don't make 1 ml का solution. Okay, we don't make. अभी मैंने mention किया, we don't make it, right? So what does it mean? So we make a stock solution to 22 microliter of the sample, and it says that it should be 50 nanogram per microliter. So in the spectrophotometer at 260 nanometer, as you know, so basically we don't use spectrophotometer for DNA concentration check. There's something called nanodrop. Okay, we use nanodrop. क्योंकि 1 ml में सोल्यूशन बनाऊंगी तो मेरा डीएनए इतना वेस्ट जा रहा है, ठीक है, हमें इतना सारा डीएनए नहीं मिलता है. सो वी ऑलवेज गो विद द नैनोड्रॉप वेयर द 1 माइक्रोलीटर रीडिंग यू गेट. ओके, आप 1 माइक्रोलीटर ऐड करते हो एंड एट 260 नैनोमीटर यू गेट द रीडिंग. सो दैट रीडिंग शुड गिव यू दैट एटलीस्ट 50 नैनोग्राम होना चाहिए उसमें, नॉट बिलो दैट. Okay, is this much clear? So this is an ideal case. Like at 260 nanometer, your 1 microliter should have 50 nanogram of DNA. This is minimum, and it means the absorbance of 1. It's clear? Now, if you have protein and phenol, it is going to affect this estimate. Why protein? क्यों? See, 260 है, protein 280 पे रहता है. So there are sometimes a few amino acids which might overlap, and phenol of course gives the reading at 260 nanometer. इसलिए मैंने बोला था starting में, even though we use phenol for DNA purification, it should be removed completely using chloroform and the absolute alcohol. Okay, so this should be removed. अगर आपने अच्छे से नहीं remove किया है, so this is going to interfere with the reading. Guys, are you able to follow?
Yes.
Okay. Now, further UV absorbance gives you no check on the integrity of your DNA. What does it mean? It means that even if your DNA is broken, okay, the machine is not going to tell you whether the DNA is intact or it is broken. ये नहीं पता चलेगा. Okay, it can be completely degraded and still give you a reading. So precise quantification can be done by running DNA or RNA on gel electrophoresis. So you know what is age, correct? It is agarose gel electrophoresis. So after this, we will be doing agarose gel electrophoresis, where you can check whether the DNA and RNA is intact or not. Why? Because you know that if it is linear or whatever, linear or circular, if it is proper, if it is intact, then only the ethidium bromide can get into between, intercalate between the base pairs, and gives you the absorbance when you check under the UV radiation. Agar it is broken or degraded, then of course the ethidium bromide cannot get intercalated, and hence no reading will be there. Okay, so that is one point. So you cannot tell about the DNA RNA integrity by checking only the UV absorbance. Now Like protein contamination or the RNA contamination can be estimated at absorbance of 260 and 280. So if you take 260 to 80 ratio, if it is high, it indicates RNA contamination. If the ratio is low, it means protein contamination. Okay. So high हम किससे लेते हैं? There is a unique say हमने calculate करके रखा है, तो machine will tell you if this is high, the ratio you don't have to do separately. आप जैसे एक बार run करते हो machine में, if I get a video, I'm gonna show in the next class. So if you put 1 microliter in the sample, just ऐसे एक dot होता है. So there is a machine which just touch this dot and gives you the everything. So it will tell you at 260 nanometer what is the concentration, what is the 260 by 280 concentration, and what is the 280 by 260 ratio. So it will tell you how much RNA and how much protein contamination is there. Okay, if it is less, I think if it is around 0.08, ऐसे कुछ नीचे है, then we can say that the DNA is good to carry out the further experiments. Clear? I know so much of details have given, and you need to remember all this so that you can do your section C of the exam. ठीक है? Confused नहीं होना है. Just listen 2-3 times and you will be done. So this is all about ATBR, which I already said. Like we use for detection and quantifying nucleic acid. If it is intact, then ATBR can get intercalated. If it is not, then it will be washed off with the buffer. Okay. So this is the most widely used method for staining the electrophoresis gel. अब थोड़ा recent में क्या हो गया? Since we know that ATBR is carcinogenic in nature. ठीक है? And people are, थोड़ा सा, sometimes they don't wear the gloves and all. So they have started using cyber green. Okay, so there are many other are there in order to label the DNA. So the fluorescence, the intensity of fluorescence will also tell you the concentration which runs on the gel, but it is always good that you go for the nano drop. Okay. Yes. Yeah, so that's all for today, guys. Any doubts? Do you want to share? Ask. कुछ पूछना है? So this is the complete DNA RNA purification, and in the next lecture I'll show how like what exactly steps you need to follow. अभी आपको पता है why we use alcohol, why we use phenol, okay, why we use alkaline conditions, why we use low salt and high salt. अभी आपको ये सब पता है. Now in the next lecture we'll discuss कब कौन सा step करना है. ठीक है? तो पहले phenol आना चाहिए, पहले alcohol आना चाहिए, क्या आना चाहिए. That's like each step wise we are going to discuss in the lab. जो आपका जो कंपनी का जो किट रहता है, that we are going to discuss. Okay, and at the end I'll show you how the nano drop works. Clear? Done? Okay.
Yes, ma'am.
Okay. Is there anything else which you want me to add in the next lecture, or it is not clear?